So good morning. So I'm presenting you today our project Microvaxxine, how to turn microbes into our allies for cancer vaccines. So who we are. So basically we are a future spin-off of the National Cancer Institute in Naples. We are focused on cancer vaccine, so basically on therapeutic cancer vaccines based on microbe-derived antigens. We have more than 10 years experience as a group. Our lead focus is HCC, primary liver cancer, but we have also secondary indications. These are the funding that we have received. These are our fundings that we have received through the years. We have experience in conducting phase 1 clinical trials, and we have 2 patents filed. So this is just to give you a flavor of the major international collaborations that we have had in the last years. 2 major projects funded by the European Union. I was the PI, and these are the total fundings that we have received, both in liver cancer, cancer vaccine development. So this is our executive team, which is a local team, me together one of my collaborators at the Institute, and this is a colleague always at the Institute as a medical oncology, but we also have already a SAB made by high standard worldwide collaborators in cancer immunology. So looking at the total addressable market, so basically we are looking at the cancer vaccine market and the liver cancer therapeutic market. And as you may very easily see, the CAGR for both of them are very important. Here we have predicted for in the next 5 years 10%, and here we have predicted 18% in the next 5 years. So which is the target product profile that we are talking about? So the indication is a therapeutic treatment of cancer patient, and the target patient population are patients affected by primary liver cancer, hepatocellular carcinoma in stage C. So what are we looking for? Very good efficacy and outcome. Basically, we want to improve at least 25% the overall survival compared to current treatment, standard of care. We want to have a very good safety, very good tolerability, very user-friendly handling and patient administration, limited adverse effects, and low to zero pre- and post-treatment medical intervention, therefore, a very high efficiency risk-benefit ratio. But let's go to the concept, to the background. So, which is the current status of the cancer vaccine development? Cancer vaccine development are based on 2 different antigens: tumor-associated antigens, tumor-specific antigens. The tumor-associated antigens are normal self-antigens, not mutated, expressed at a very low level on normal cells, but they are overexpressed on cancer cells. Tumor-specific antigens are antigens not expressed on normal cells, but expressed only on not expressed on normal cells, but expressed only on cancer cells. Therefore, we can make off-the-shelf vaccine with these ones, and only personalized vaccines with the TCA. We want to focus on tumor-associated antigens. We want to make off-the-shelf vaccines, vaccines for all patients affected by that tumor. Indeed, they are shared antigen, as I said, so we can make off-the-shelf vaccine. The cost is very limited, but we have to face 2 major hurdles. The immune tolerance, because being self-antigens, the immune system is not willing to respond against these antigens, and obviously we need to show that they are efficacious. At this moment, only 2 vaccines have been approved by FDA based on tumor-associated antigens. Actually, these are the only 2 cancer vaccines approved for any kind of antigen. So how do we circumvent the immunotolerance? This is our solution. So basically, we have found in several models that antigens derived from microbes, namely viruses and bacteria, can be very similar to tumor-associated antigens. Not only in the linear sequence, you may see the green residues, which are identical, but also in conformation, when we compare the microbe-derived antigen and the tumor-associated antigen. So this means that when we inject, or when we are going to inject in patients in non-self antigens very similar to tumor-associated antigens, we will not face any immunotolerance. We will elicit a very strong immune response, and this immune response is going to cross-react against the tumor-associated antigens. So in this way, we will not eliminate the problem of the immunotolerance. And indeed, that's what we showed in ex vivo studies. So basically, we showed that indeed T cells are able to cross-react with the microbe-derived antigens and the homologous tumor-associated antigens, and T cells elicited by the microbe-derived antigens are able to kill cells presenting tumor-associated antigens. So cross-reactive immune response. Payoff: overcoming immune tolerance and low efficacy of shared self-antigens. Highly homologous non-self microbe-derived antigens potently induce T cells cross-reacting with tumor-associated antigens. But there is also an additional advantage. These microbe-derived antigens are similar to tumor-associated antigens overexpressed by several tumors. So this means that we can hit with the same vaccine based on microbe-derived antigens several tumors. That's what we call one vaccine for many tumor strategy. And this is just what we are envisioning. A vaccine based on microbe-derived antigens for several cancers, T cells able to kill several tumor-associated antigens, cancer regression in several tumors. So this is our pipeline and platform. So basically, we search for tumor-associated antigens overexpressed by tumors. We check for homology between the tumor-associated antigen and the microbe-derived antigens. We validate by immunological means, and we develop vaccine for phase 1 clinical trial. As I said, now we have 2 patents filed for antigens derived from viruses with the similarity to tumor-associated antigens and antigens derived from microbes. The competition. Well, I cannot show you just a very limited number of competitors because the field is very well populated by competitors. But they are mostly universities, mostly very small companies. But I want to show you that if we look at the cancer vaccine overall, even though we are talking about the 360 cancer vaccine trial right now conducting, most of them are in a very early stage of trial and only 2% in phase 3. But if we look at the cancer vaccine for liver cancer, for hepatocellular carcinoma, there are only 13 trials undergoing, but none in phase 3. So the field is wide open. So which is our state? I mean, where are we now? So basically, we have one candidate vaccine, what we call MIVA-001, focused on liver cancer. We already have the vaccine prepared in GMP. We already have the vials in GMP prepared, and we are going to perform a phase 1 clinical trial in 2025. Then we have already a MIBA-2 candidate, which will be a multiple basket trial. In 2025, we will have a candidate selection, IND preparation 2026 and 27. For these, obviously, because we are talking about clinical trials, our seeding funding that we are looking for is about 5 million. And in 2025, for the first year, if everything goes as we hope, obviously, this will be scaled up significantly up to something about 12 million. Obviously, we do have a business exit strategy, which includes acquisition by another company or license agreement with another company. And obviously, we are thinking about the major players in the field of cancer vaccine. So in summary, we want to develop an off-the-shelf vaccine based on highly innovative microbe-derived antigens for overcoming immunotolerance and poor immunogenicity of tumor-associated antigens. Our vaccine, MiVA-001, has been already manufactured in GMP for testing in phase 1 clinical trial. The lead focus, as I showed you, is a primary liver cancer, which has a high level of unmet medical need. The numbers are about 1 million deaths per year in the world today, and is considered the third cause of death by cancer overall in the world. But we are proposing a one vaccine for many tumors, therefore we will have multiple secondary cancer indications. As I showed you, it's very important CAGR for both cancer vaccine and liver cancer. The process of production is very established, and there is a long period of storage, basically more than 6 years when the vaccine is manufactured, very high cost-benefit ratio, user-friendly handling and patient's administration, and finally, very high patient's adherence, no need for hospitalization, no major adverse events. Thank you so much.